Kuzuzangpo, welcome to Bhutan e-learning program. This is a biology lesson for Key Stage 5, that is for classes 11 and 12. I am Mahendra Timsina from Deshincholing Higher Secondary School. Before I begin with the lesson today, let us look at some words that we have been talking about in our classes. The first one, it is reproduction. Second one, procreation. And the third one, it is cell theory. When you look at the three words, it gives us a sense that we are talking about the formation of one organism or the production of new individuals from the parent organisms. The second thing that I want to talk here is we know a male produces millions of sperms. However, only one child is born. Have you ever wondered why? So, to understand that, today in this lesson, we are going to talk about fertilization and some events after fertilization, that is, the post fertilization events in human reproduction. At the end of this lesson, you should be able to define fertilization, relate the types of gamete to the sex of the child born, and third one, you should be able to explain various types of activities or changes that occur during implantation. Now let us move on with the lesson. We know there are two different types of gametes, or we call that as sex cells. In males, spermatogenesis occurs. Because of spermatogenesis, sperm cells are produced. The sperm cells are haploid and they contain 23 chromosomes. Similarly, in female, oogenesis occurs, leading to the production of female gamete. Female gamete, just like the male gamete, also contains 23 chromosomes. So both are haploid. They fuse during fertilization and lead to the formation of zygote, which is a diploid cell. It contains 46 numbers of chromosomes, or let us say 23 pairs of chromosomes. To add something more, we represent male as XY. And during spermatogenesis, a male produces two types of gametes, that is X type and Y type, whereas female is represented as XX, and they produce only one type of gamete, that is X type. So during fertilization, if X of the male gametes fuses with the female gametes, then it will lead to the formation of a zygote, which is XX. That will lead to the formation of a daughter. Whereas if the Y gamete fuses with the female gametes, then it will form a son, or it will lead to the production of a son. So that was all about the general representation of the types of gametes. Now, before we move on, it is also important for us to recollect about the gametes. So if you look here, this is the male gamete, which we call as sperm or the spermatozoa. So it has different parts: the head part, the neck, the middle piece, and the tail. Out of all, I would like to focus on is the head part. If you look at the head part, there is an acrosome. It is really important because it contains enzymes which help during the process of fertilization. And the second important thing here is the nucleus, which contains the chromosomes coming from father. And the second diagram here, it is the female gamete, that is ovum. If you look at the ovum, you see there is a nucleus, ooplasm, then a membrane, that is the vitelline membrane, zona pellucida, and the corona radiata. The cells of corona radiata are connected by one acid which we call as hyaluronic acid. We also should know the female reproductive system. So you have on the screen the female reproductive system. So if you look at the female reproductive system, it has certain parts. So let me specify them. The vagina, cervix, the uterus, fallopian tube, and ovary. So from all, we should focus on ovary because oogenesis occurs inside the ovaries. Uterus, the inner wall of the uterus is called as endometrium. It is where embryo later attaches and undergoes the embryological changes. Now, let us start with fertilization. First thing is insemination. We can define insemination as deposition of semen into the female genital tract. During copulation, semen is deposited into the female vaginal tract. So during this, about 2 to 300 million sperm cells are present in the semen. Normally, about 3.5 ml of semen is deposited. We should remember, semen is not just the sperm cells, but also contains the secretion of other glands, which we call as the accessory glands. So after insemination, the second step is movement of sperm. We know fertilization occurs in some part of the fallopian tube. So after insemination, the sperm cells move from the vaginal tract to the ampulla where fertilization occurs. Sperm cells move by lashing movement of their tails. However, it may not be enough. That's why there are two hormones, that is oxytocin and prostaglandin. They cause the contraction of the muscles in the female reproductive system, and the movement of those muscles also help in the sperm movement. The other thing is, all the sperm cells deposited in the female genital tract may not reach the area of fertilization because of many factors. Maybe because of acids there, or maybe because of some physical deformities. It is also important for us to know that after deposition into the female genital tract, the sperm cells survive for around 72 hours. The next important thing that is occurring simultaneously is the arrival of ovum. In the ovaries, the ovum from the ovary is received by the infundibulum, and the ovum travels to the fallopian tube. And there, the ovum also survives for about 72 hours, which means after ovulation, the fertilization has to occur within the 72 hours. The fourth step is capacitation of sperm. The sperm cells, as deposited during the sexual intercourse, are not fit for fertilization because they are surrounded by some factors which we generally call as the decapacitation factors. So in this step, the decapacitation factors are removed. So during the capacitation process, as you can see in the diagram, a lot of cholesterol vesicles, some antigens are removed from the sperm head. Apart from that, the sperm head is also made more permeable for the entry of ions such as calcium because they help during the process of fertilization. And capacitation also enhances the movement of sperm cells. So after that, the sperms reach the area of fertilization. Now after they reach there, the sperm cells come in contact with the ovum. But what we should remember here is not all the sperm cells which reach the area of fertilization are going to fertilize the ovum. Out of many, let us say out of thousands which reach there, only one of the sperms is able to fertilize. The next step is called as acrosomal reaction. What we can see is a sperm cell has come in contact to the outermost layer of the ovum, and then it is releasing the enzymes, and the enzymes are bringing in some changes, opening the path for the sperm to enter, and then the sperm is entering. So acrosomal reaction here means the changes that is brought about by the enzymes that are present in the acrosome. Once the sperm comes in contact with the corona radiata, the acrosome opens up and the enzymes in the acrosome are released. There are enzymes like hyaluronidase, corona penetrating enzymes, zona lysins or acrosins. The first enzyme, hyaluronidase, it breaks down the hyaluronic acid which connects the cells of corona radiata. It makes the cells free, creating a loose environment. And the corona penetrating enzymes will break down the cells, creating the path for the sperm. And that will lead or help the sperm to move in. Apart from that, there is also a check of compatibility. There are factors called as fertilizins and antifertilizins which keep a check on whether the sperm is compatible. After the acrosomal reaction, sperm entry occurs. The sperm cells reach the zona pellucida, and the enzymes zona lysin or acrosin breaks down a small portion of zona pellucida in that area where acrosomal reaction occurred and creates path for the sperm to enter. After the sperm entry, the next one is cortical reaction. So right after the zona pellucida is broken, the plasma membrane of the sperm and the ovum fuses, which means the ooplasm comes in contact with some cytoplasmic material of the sperm. So when that happens, on the outer side or the outer part of the ovum, there are some granules called as cortical granules. They contain enzymes. So once the fusion of plasma membrane of the sperm cells and the ovum occurs, the cortical granules will move towards the plasma membrane and they will fuse with the plasma membrane. When they fuse it, the enzymes present inside them is deposited in the space which we call as the perivitelline space. So these enzymes are really important because once the sperm has fused, they will cause hardening of the zona pellucida. Apart from that, outside the zona pellucida, the corona radiata cells will rearrange, and also the sperm cells which are now outside, they are made incapable of fertilization by incapacitation. So as a result, only one sperm cell has entered inside and delivered the male pronucleus into the ovum. After the cortical reaction, the next important step is activation of egg. Even after ovulation, the ovum is not ready for fertilization. It is because it has not completely undergone the process of meiosis. The female pronuclei is not completely divided. The ovum, the cell inside, is in secondary oocyte stage. That's why only after the entrance of the sperm, the meiosis completes and the secondary oocyte will divide and it will form the female pronuclei which can now fuse with the male pronucleus. So that's activation of egg. After entry of sperm cell, the meiotic division in the ovum is completed. The last step for fertilization is amphimixis or karyogamy. In simple terms, we call that as fusion of the nuclei, that is the female and the male nuclei. Firstly, as the sperm enters, it enters through a path which we call as the penetration path. It may not be lying exactly in the location where the female pronucleus is located. That's why the male nucleus can change a course, change a path. So the new path is formed which we call as copulation path. The copulation path enhances or helps the male pronucleus to move to the female pronucleus. So it is the path followed by the male nucleus. And fusion of the two nuclei will occur, resulting to the formation of a bigger diploid nucleus, which we can call as synkaryon. This process commonly can also be called as syngamy. So that was all about fertilization. Now let us move to post-fertilization events. I have again on the screen one diagram, a picture that shows various activities that occurs after fertilization. You can see fertilization on the day 0, then zygote, and then as you go, you see there are changes occurring. As the zygote continually divides, it also moves from the oviduct towards the uterus, where implantation occurs. Right after fertilization, the zygote is formed, which is a diploid cell. Zygote undergoes a fast process of division called as cleavage. During cleavage, the zygote undergoes first division forming 2 cells, then second division forming 4 cells. Likewise, it undergoes some division reaching to 16 to 32 cell stage, which we call as morula. So cleavage starts from zygote and ends with reaching the stage called as morula stage, and still it is covered by zona pellucida. So after cleavage, the second step is called as blastulation. Morula, which is 16 to 32 cell stage, undergoes division as well as differentiation. During the change, the morula cells which were undifferentiated now arranged to form 2 layers, the outer layer of cells and an inner mass of cells. The outer mass or the outer layer of cells is called as trophoblasts, whereas the inner mass of cells are commonly called as inner mass, or we can even call them as the embryonal cells. They arrange as given, they get arranged like shown in the picture. The outer mass of cell covers everything, and the inner mass of cell will move to one side, and a big space is created, which we call as blastocell. More or less at that stage, the blastocyst has reached the uterus. So before we move on to the next stage, let me ask you a question. What helps the zygote to move to the uterus as it is dividing? I guess you got something right. The inner layer of the fallopian tube contains small finger-like projections, which they are like cilia which keep on beating, and as they move, they push the growing embryo towards the uterus. So towards the last stage of blastocyst, or towards the 8th day, you see the blastocyst has reached the uterus. After reaching the uterus, the next step is called as implantation. Implantation here means the entering, or let us say attachment of the blastocyst to the endometrium, that is the inner part of the uterine lining. Now, let us watch a small clip to understand the process of implantation.
Implantation of the blastocyst usually occurs 6 to 8 days after fertilization. By the end of day 8, the blastocyst has burrowed into the endometrium of the uterus. At this time, it is composed of 2 main components: the outer cell mass, the trophoblast, and the inner cell mass, the embryoblast. As the trophoblast makes contact with the endometrium, it differentiates into 2 layers: an inner cytotrophoblast and an outer syncytiotrophoblast. The embryoblast differentiates into a bilaminar embryonic disc composed of 2 cell layers, the hypoblast and the epiblast. Soon after the embryonic disc has formed, a cavity begins to appear between the epiblast and the cytotrophoblast, known as the amniotic cavity. Cells originating from the hypoblast begin to migrate, forming a thin membrane, which covers the inner surface of the cytotrophoblast. This is called the exocelomic membrane. The exocelomic membrane and cells of the hypoblast together form the walls of the primitive yolk sac. By day 9, the blastocyst is completely embedded in the uterus wall. At this stage of development, the growth of the syncytiotrophoblasts and cytotrophoblast is much quicker than the bilaminar embryonic disc. Small holes called lacunae begin to form in the syncytiotrophoblast as it continues to expand. By day 12, the lacunae stop growing and fuse to form large interconnecting spaces called lacunar networks. Capillaries in the endometrium surrounding the developing embryo dilate, forming maternal sinusoids. As the syncytiotrophoblast continues to expand, enzymes begin to erode the lining of the sinusoids and uterine glands, allowing maternal blood and uterine secretions to flow into the lacunar networks, establishing a uteroplacental circulation. The blood and uterine secretions only come into close proximity to the embryo, allowing the exchange of gases and metabolites.
So how was the clip? We saw some important things in the clip. First, we saw that the zona pellucida is removed before the blastula enters into the uterus or the endometrium. There were some changes. So, as shown in the video, as the blastula came into contact with the walls of the uterus, there was cell division, release of some enzymes, due to which some path was created into the endometrium. And the blastula totally enters into the endometrium, and after entering, it is covered. As the blastula entered the endometrium during implantation, the embryonal cells also undergo some changes. They divide and they undergo the structural changes which we call as differentiation. So that was implantation. Now after implantation, there are some important steps. I will try to highlight on them. The next step is called as gastrulation. So after the embryo has got implanted, the embryonal cells undergo division, and during gastrulation they form three layers called as ectoderm, mesoderm and endoderm. These three layers later will lead to the formation of different organs of our body. After gastrulation, another important activity is called as neurulation. Neurulation is actually marked by the formation of neural tube, which later leads to the formation of our nervous system. After neurulation, then gradually other organs start to develop. So we call that as organogenesis. So the entire process takes about 9 months, starting from fertilization till the formation or full development of the baby. It takes 9 months. That's all for today's lesson. So before we leave, let me leave you some questions for you to explore. The first one, we saw pregnancy, or let us say the development of child, or the gestation period had a lot of changes within the female. So a lot of care has to be taken. So can you think and make a list of what type of care do we need to give to the women during pregnancy? Second important thing is a lot of cell divisions were occurring, a lot of morphological changes were occurring. So in that time, a good diet is important. So can you do a small research and design a plan, a diet plan for the pregnant lady during the to be provided during the pregnancy period. That's all for today's lesson. Thank you and stay safe.